Hello, good morning, everyone. My name is Ran Wang. I'm a PhD candidate in plant breeding group, Wageningen University. So today I'm going to present my PhD project. The title is Genetic Mapping and QTL Analysis of the Sex Locus in Tetraploid Kiwiberry. This is a collaboration project with Zhengzhou Fruit Research Institute of Chinese Academy of Agricultural Science. Usually we see there is fresh kiwi fruit in the market, brown skin with hot hairs. Recent years, there's a new type of kiwi fruit has been commercialized as kiwiberry. So this kiwiberry is smaller than kiwi fruit. The skin is soft and smooth and hairless. Kiwi fruit and kiwiberry, they belong to the genus Actinidia. And the kiwiberry, that's the common name for the species Actinidia aguta. It takes usually 3 to 5 years to flower. So to breed a new cultivar, it usually takes 10 to 15 years. Kiwiberry is diverse in purity level. It has a similar taste as kiwi fruit, but size is smaller. It's like a grape size. The skin is soft, hairless, and edible. So we can eat a whole fruit. That's perfect for snacking on. It also is a good source of fiber and vitamin C. Almost all Actinidia species are functionally dioecious. So the male and female flowers are on separate plants. Only female flower produce fruits. There are some challenges in kiwiberry breeding. It's dioecious. That means for the female plants, we know the fruit phenotype, but the male plants, we're not sure, 100% sure when we set up a cross. So it's like a blind choice for the father. And the kiwiberry is a polyploid. That makes the genetics a little bit complicated. And the genome is highly heterozygous. It has 29 pair of chromosomes. So this makes the kiwiberry breeding a little bit challenging. At the moment, the conventional breeding still use the selection is still based on the phenotype. So the many cultivars are direct select from the well materials or just selected from the first generation of the cross. So that take a bit, the breeding is time consuming and also the cost consuming. So we hope to carry out the research to develop the molecular breeding tools that we can use to speed up the breeding process and also to increase the breeding efficiency. So here is the aim of my project is to carry out the genetic mapping and QTL analysis of important traits in the tetraploid kiwiberry population. The material we use is a tetraploid biparental population, consists of 315 F1 progeny. This is the female parent of the maple population. So the fruit is red skin and red flesh. The project involves to develop a SNP array for the kiwiberry and to construct a linkage map for the maple population and to carry out the QTL mapping of the sex locus in the kiwiberry population. For the SNP array design, we use the Affymetrix enzyme platform. we sequenced a set of kiwi fruit accessions, including the parents of the mapping population. The array design involved a number of routine steps for the array design. So here I just highlight a key step that is very useful to make the array work. The kiwiberry, we don't have the reference genome. We use the diploid kiwi fruit genome and modify it to be more specific to the kiwiberry sequence, so we can use it to design the probe to design the probe. So the final array contains 135K SNPs. We use the array to genotype the mapping population. The marker dosage were called using FigPoly. There are 118K usable markers representing 24 marker types. And the markers were processed in PolyMapR. After some quality check, such as filter out the markers with high missing value, and some markers did not show the expect segregation to the parental population, to the parental dosage, are also removed. And some markers with the same recombination frequency are marked as duplicate markers, so that we reduce the number of markers to 77K markers, and these will convert into 9 simplest marker types. And we use this dataset to construct the linkage map using PolyMapR. The kiwiberry has 29 chromosomes. We use simplex, nulliplex, and nulliplex simplex markers for the initial clustering of 116 homologs. And these homologs were bridged using duplex, nulliplex, and simplex markers that covers all 4 sets of 29 chromosomes. And then the other types of markers were subsequently assigned to a linkage group using the linkage to the simplex, nulliplex markers. And then we added back the duplicate markers to generate integrate linkage map. And from this figure, we the map we don't see a lot of tensions and gaps here, and it looks it's a good quality map. So we extract marker and map to the kiwi fruit genome. And some linkage groups were reordered and according to the physical map that produced the final linkage map here. We further compared the genetic position of the marker against their physical position of kiwi fruit genome by merit plot. And from the figure, it shows a high degree of synchrony between kiwiberry and kiwi fruit genome. And from the merit plot, we can see the chromosome arms and the centromere are very clear on the plot. So it also shows that our linkage map is a good quality map. The kiwi fruits, the sex system in kiwi fruits is XY system. So for kiwiberry, the tetraploid kiwiberry, that will be XXXY. We estimate the IBD probability in the population using QTL. We found a major effect QTL associated with male sex on linkage group 3. It's a simplex QTL. The segregation type is here. This big Q is the allele for the male sex. We check the markers within the QTLA region. So some markers are male-linked markers that can be useful to determine the sex in the seedlings in the breeding program. So finally, we investigate the meiotic pairing behavior in using the IBD probabilities. Under assumption of bivalent pairing, we apply a chi-square test for deviation from the expect counts, assuming the random pairing. Here the figure, the homologs from mother are labeled as A, B, C, D, and homologs from father labeled as E, H, GH. So the thickness of the line represents the deviation from a random pairing model. If the pairing, more pairing are labeled as red, less pairing is colored as blue. So here, from this figure, all combination possible pairing are shown in the figure. We observed that this linkage group 15 of the male parents that is significantly deviates from random pairing. And we also can see some chromosome. The pairing happens more often systematically in both parents. So here we conclude the kiwiberry has mostly polysomic inheritance with preferential pairing for some chromosomes in our population. So here's the conclusion. We generate a high density linkage map in kiwiberry population. And our genetic mapping showed that there's a high synchrony between kiwiberry and kiwifruit genome. And the kiwiberry has mostly polysomic inheritance with preferential pairing for some chromosomes. And these results are wrapped up in a publication on Plant Biotechnology Journal. Before end of my presentation, I want to thank my supervisor, Chris Marliquat from Plant Breeding Group, Wageningen University. I want to thank Rachel, Peter, Paul, Danny, and Julian. Thank you for your support. And I want to thank my supervisor from CAS and my colleagues from kiwiberry, kiwifruit breeding team. So thank you, everyone.